Good day everyone, myself Aradhya Singh and along with me are my friends Adesh Tiwari and Aditya Raj. We study in Ramsher Thakur Public School in class 9th A and together are known as the DNA scientists. Today I will take you through a fascinating experiment, DNA extraction from fruits. Before that, let us understand what DNA actually is. DNA stands for deoxyribonucleic acid. It's the genetic material of what an organism contains inside their nuclei. DNA contains instruction that makes an organism unique. It helps scientists study genes, identify organisms, and make up new medicines. Now let us talk about why do we extract the DNA. We extract DNA to study the genetic makeup of an organism. It helps in forensic science, genetic tests, and disease research. It is used in agriculture to create better disease resistant crops and is also important in medicine, vaccines, and treatments. Now let us understand what are the things required for this experiment. The first and the basic thing is fruit. It can be any type of fruit which is easy to mash, for example a tomato or a banana. The next thing that is important is isopropyl alcohol. This alcohol makes the DNA visible. It is white, cottony strands floating on the top of the liquid. Now let us understand the steps required for this experiment. The step 1 is cell lysis, which means mashing the fruits basically. In this step we mash the fruits to break their cell open. The step 2 is protein denaturation. In this step we add a extraction buffer for removal of proteins. Step 3, filtration. In this step we filter out the solid fruit parts from the DNA rich solution. Step 4, DNA precipitation. In this step, we add the alcohol into the DNA-rich solution. This makes the DNA visible. It is white, cottony strands floating on the top of the liquid. Step 5, spooling. In this step, we take out the DNA using a spool or a forceps. It is white, cotton-like structure. Now let us talk about why this experiment is important. It demonstrates that DNA is present in all living organisms. This method is simple yet shows real biological science in action, forms the base of research in genetics and medicines. DNA extraction converts classroom science with real world discoveries. It is a wonderful example to show how biology and chemistry work together to make a fascinating form. Thank you.